Hello, my name is Kelsey Stewart, and I'm a member of the DeMail Research Group in the Department of Chemistry. Today, I'll be sharing with you some of our work merging whole cell biocatalysis with organocatalysis to upgrade alcohol feedstocks in a one-pot reaction. Let me begin by briefly touching on the motivation for this work. As we strive to curtail global warming, one area we can look to is industrial manufacturing. In 2017, the EPA reported that industry is responsible for 22% of total greenhouse gas emissions in the US. Included in this sector is the production of commodity chemicals, with the production of petrochemicals resulting in the release of nearly 60 million metric tons of CO2 each year. This is shown by the red line at the top of the plot to the right. The processes used to make these chemicals are often highly energy intensive, requiring high temperatures and pressures, multiple separations, and often result in hazardous waste streams. As a result, researchers are increasingly turning to bio-based processes to help mitigate these effects. Bioprocesses can act to minimize energy and waste streams due to their mild temperatures, aqueous conditions, propensity for renewable substrates, and one-pot nature. Additionally, it can be beneficial to use whole cells over isolated enzymes, as whole cells are able to regenerate cofactors, negate the need for costly isolation, and provide a self-replicating catalyst. However, the range of native metabolites lack the ability to meet commercial demand. In order to expand upon native abilities, researchers often use genetic engineering. Despite immense advancements in the field over the past decade, rewiring the metabolic network within an organism remains a challenge. To illustrate some of these challenges, let's look at a simplified example. Say you want to make product C from substrate A, which proceeds through the intermediate B. However, the conversion of A to B proceeds faster than B to C, which leads to a buildup of B, and a portion of B is then redirected through the parasitic side reaction to make unwanted byproduct D. To mitigate this, you inhibit the B to D pathway and upregulate B to C. Unfortunately, this results in feedback inhibition in the A to B pathway. However, despite the inherent complexities, genetic engineering will remain an important tool when designing biosynthetic processes. Further supplementing the strategy with biocompatible chemistry, that's the use of non-enzymatic reactions to influence metabolism by chemically modifying small molecules in the presence of living organisms, will only act to further expand the scope of biosynthetic processes. We can turn to adjacent fields for examples that interface microbes with non-enzymatic chemistries. Take, for example, bioelectrochemical systems. Bioelectrochemical systems are the most well-established one-pot microbial system. In a bioelectrochemical system, microbes interact directly or indirectly with the working electrode to accept or donate respiratory electrons. Electrons donated to the electrode are used to generate bioelectricity, and electrons accepted by the microbe are used to fuel metabolic activity. More recently, the working electrode has been used to alter the oxidation reduction potential of the system, which can redirect flux towards more desirable products in a process called electrofermentation. Along the same lines, researchers are working to realize artificial photosynthesis. A recent strategy uses photoactive nanomaterials that are assembled onto the cell surface or taken up by the cells. These materials transfer photogenerated electrons to the cell to fuel the metabolism of carbon dioxide. While both fields can provide valuable insight for working with hybrid systems, they focus mainly on energy conversion and not the expansion of product scope. For that, we can turn to examples in which inorganic catalysts are used to further upgrade fermentation products or even bridge gaps between metabolic pathways. As the field of biocompatible chemistry grows, we see more examples illustrating the benefits of merging biotic and abiotic chemistries. Yet, the use of organocatalysts has remained largely unexplored. Our work seeks to address this. By leveraging the broad reactivity and substrate flexibility of organocatalysis, we can continue to build on the number of products accessible through bio-based processes. We sought to design a one-pot process that further upgraded bacterial metabolites through a concurrent organocatalyzed step. The biocatalyst we chose to work with is the bacteria Glucanobacter oxidans. Deoxidants have a unique oxidative metabolism capable of converting alcohols to their corresponding aldehydes. The enzymes responsible for this conversion are located in the membrane with the active site oriented toward the periplasm. That's the space between the inner and outer membranes of a bacterial cell. This means that the alcohol substrate never fully enters the cell, and the aldehyde product is expelled into the media. We chose to explore a number of biogenic amines as potential organocatalysts. Previous work has shown that these amines are capable of catalyzing aldehyde addition condensation reactions in aqueous conditions and are well tolerated by microbes. The target product is 2-ethyl-2-hexenal, a direct precursor to the high-volume chemical 2-ethylhexanol, which is a common component in many plasticizers. The overall design features a biphasic system with an aqueous and organic layer. In the aqueous layer, the butanol substrate is oxidized to butyraldehyde by G-oxidants. The butyraldehyde undergoes a self-alkyl condensation catalyzed by the biogenic amine to yield 2-ethyl-2-hexenal. This partitions into the organic layer. The use of an in situ extractant helps to minimize toxicity of the product to the microbe. To start, we screen the biogenic amines to determine which were most effective in the integrated system. The G-oxidants were suspended in phosphate buffer with an optical density of 1. We used a 1:5 ratio of the extractant isooctane, a starting butanol concentration of 5 milligrams per milliliter, and 250, 500, or 750 millimolar of each catalyst. Samples of the isooctane layer were taken at 6, 24, and 48 hours, and the 2-ethyl-2-hexenol was quantified with gas chromatography mass spectrometry. The most effective catalyst concentration for each amine was repeated in triplicate, and the results of that study are illustrated in the graph you see here. Each cluster of 3 bars represents the percent yield of 2-ethyl-2-hexenol at 6, 24, and 48 hours. The catalyst, its structure, and the concentration used are denoted below the x-axis. The catalysts from left to right are glycine, lysine, 3-amino-propanoic acid, 4-amino-butanoic acid, 5-amino-pentanoic acid, and 6-amino-hexanoic acid, and proline. From these results, 3 catalysts stood out. Glycine because it reached close to maximum conversion after only 6 hours. 3-amino-propanoic acid because it reached the highest overall conversion. And lysine because it showed comparably good performance at the lowest catalyst loading and can be sourced inexpensively. These 3 catalysts were explored further by varying the initial alcohol concentration. Initial butanol concentrations of 2, 5, 7, and 10 milligrams per milliliter were studied. Again, samples were taken at 6, 24, and 48-hour time points. The data points represent averages of 3 trials plus or minus 1 standard deviation. An initial butanol concentration of 10 milligrams per milliliter gave poor yields for all catalysts. Glycine and 3-amino-propanoic acid reached maximum conversions of around 60% for 7 and 5 milligrams per milliliter butanol, respectively, whereas lysine reached conversions of greater than 70% with the lowest alcohol concentration of 2 milligrams per milliliter. In addition to achieving good yields of the target product, we were able to show that non-enzymatic catalysts can be used to redirect flux within a system. At extended processing times, G-oxidants will over-oxidize butanol to its corresponding carboxylic acid, butanoic acid. The addition of lysine acts to capture the aldehyde intermediate, redirecting flux to 2-ethyl-2-hexanol with 37% yields. The addition of the biocompatible extractant further increases this yield to 70%. Additionally, I briefly mentioned previously that lysine was a good candidate because it could be sourced inexpensively. Lysine is mass-produced as cattle feed. Utilizing cattle feed lysine as our catalyst did drop yields to 57%. However, it also reduced catalyst costs to less than 1 cent per gram. To round out our study, we explored the substrate scope of our optimized G-oxidant lysine system. C2 to C6 primary alcohol substrates were used to generate self and cross-aldyl condensation products. Both N-propanol and N-pentanol showed good conversion to their self-aldyl products with yields of 68 and 88% respectively. Hexanol produced poor yields, likely due to the increased hydrophobicity of the substrate. Ethanol could not be captured as a self-aldyl product, but could be captured as a cross-aldyl product with propanol or pentanol. To conclude this presentation, we showed that simple alcohol feedstocks can be upgraded into industrially relevant compounds using a combination of live bacteria and chemical catalysts. This upgrading occurs in aqueous buffer at 28 degrees Celsius using inexpensive and renewable chemical catalysts. Our current work is focused on using an engineered strain of E. coli to directly form industrial products from glucose. With that, I'd like to thank you for taking the time to listen to this presentation. Thank you to the DeMail Group, my thesis committee, and the chemistry department staff for all their help and support. Thank you to the Colorado School of Mines for funding this work. And thank you to my friends and family for the continued encouragement. I hope everybody has a great day.